Mendel turned his attention to simultaneous inheritance of two or more apparently unrelated traits in bees. Mendel was asked how two pairs of alleles would segregate in a dihybrid individual. Alleles are alternative forms of a single gene. A dihybrid individual is a plant that's heterozygous for two genes at the same time. Mendel crossed yellow round peas with green wrinkled peas and obtained a dihybrid F1 generation, showing the dominant phenotypes yellow and round. Big R is the dominant or expressed trait, little r is a recessive or hidden trait. F1 is the first filial generation, progeny of the parental generation. Mendel allowed F1 dihybrids to self-fertilize to create F2 generation that could look like the parental types that were yellow and round and green and wrinkled, or the recombinant types that are yellow and wrinkled or green and round. F2 is the second filial generation, the progeny of F1. Parental types are organisms that resemble the parental generation. Recombinant types are new phenotypic combinations. One of Mendel's F2 generations yielded 315 yellow round, 108 green round, 101 yellow wrinkled, and 32 green wrinkled peas, providing evidence that shuffling of alleles of different genes took place. A gene is a unit of inheritance. A phenotype is an observable characteristic, such as yellow or green pea seeds. From these ratios, Mendel inferred the independent assortment of gene pairs during gamete formation. Independent assortment is the biological mechanism of shuffling of gene pairs during gamete formation. The presence of one allele for a gene provides no information for the allele of the second gene. Mendel understood that the different kind of germinal cells of a hybrid are produced on the average in equal numbers. Germinal cells are egg or sperm cells. Each dihybrid can make four kinds of gametes, with many gametes creating an almost perfect ratio of 1 to 1 to 1 to 1. At fertilization, 16 zygotes can be produced by four different kinds of eggs combining with any one of four different kinds of sperm. Punnett squares help us visualize this and understand that each of the 16 boxes with colored peas represents an equally likely fertilization event because each of the different gamete types is produced at equal frequency in each parent. Punnett square is a simple method for tracking kinds of gametes produced as well as possible combinations that might occur at fertilization. According to the product rule, the frequency of progeny type in each box is 1/4 times 1/4 equals 1/16. Product rule is a probability of two or more independent events occurring together is a product of the probabilities that each event will occur by itself. Some of the 16 potential allelic combinations are identical because the source of alleles does not matter. Only combination of traits is observed, we see a ratio of 9 to 3 to 3 to 1. If only at pea color/shape, then 3 to 1. For what he was trying to study, Mendel saw that there are 12 yellow per 4 green and 12 round per 4 wrinkled, a ratio of 12 to 4 or 3 to 1. This analysis became the basis of one of Mendel's genetic principles, the law of independent assortment. The law states, during gamete formation, different pairs of alleles segregate independently of each other. To find the probability that two independent events, such as yellow and round, will occur simultaneously in the same plant, the calculations would be as follows.